Now that we have succinyl-CoA, let's look at the next reaction of the TCA, which is catalyzed by succinyl-CoA synthetase. In this reaction, the high energy thioester bond in succinyl-CoA is hydrolyzed, and the energy of it is used to bring GDP and phosphate together to make GTP. And this occurs in eukaryotes, whereas in prokaryotes, it's ADP plus phosphate to make ATP. And in the process, that high energy thioester is hydrolyzed, its energy is used to make the GTP if you're a eukaryote, and the coenzyme A is released as a CoAsh, as you can see right there. And succinyl-CoA is converted to succinate. So let's look at this reaction. It's also succinyl-CoA synthetase is also called succinate thiokinase. Please know both names for this enzyme. That's important. It actually catalyzes a substrate level phosphorylation. So that's very important. It's the only reaction in glycolysis that catalyzes substrate level phosphorylation. And you're making a nucleotide triphosphate. It's GTP in eukaryotes and ATP in most prokaryotes. And GTP in most eukaryotes, but GTP is interconvertible with ATP, so you can really see it as a way of making ATP equivalents in this reaction, even if you're a eukaryote. The last thing you should keep in mind is that the synthesis of this reaction, we've actually alluded to this when we were looking at the previous slide, the energy for the reaction comes from, thus the synthesis is driven by the hydrolysis of the high energy thioester bond in the coenzyme A ester or thioester. Also keep in mind, this is also very important, the mechanism involves a phosphohistidine. So please remember that the succinate thiokinase or succinyl-CoA synthetase reaction involves a phosphohistidine. Here's what happens. Just here's the overall reaction. Succinyl-CoA, or here's what happens actually in the stepwise reaction. Succinyl-CoA plus inorganic phosphate. Inorganic phosphate can attack the carbonyl here. The coenzyme A thioester is hydrolyzed, coenzyme A is released as its thiol form CoAsh, and I end up with succinyl phosphate. If you look at this bond, what kind of a bond is it? Is it a high energy bond? Yes, because it's a mixed anhydride bond, and we know that mixed anhydride bonds are high energy bonds. So there you go. You first get a high energy succinyl phosphate bond. What happens next? The enzyme has a histidine. The histidine actually acquires the phosphate. So this high energy mixed anhydride bond right here is hydrolyzed, the histidine attacks the phosphate, and I end up with the hydrolysis of this high energy mixed anhydride bond, and I end up with succinate released. So I end up with my product, succinate, and I end up with a phosphohistidine residue where the phosphate is bound to a histidine that's part of the enzyme. A high energy, a phosphohistidine, excuse me, is a high energy bond where you have a high energy PN bond. And the energy of this is used to transfer the phosphate to GDP. The phosphate in GDP attacks the phosphate in the phosphohistidine residue. I get phosphate transfer. I end up with my histidine residue regenerated, and I end up with GTP. Of course, GTP is formed in eukaryotes, whereas this same reaction would occur, but with ADP forming ATP in prokaryotes. So now, is this a substrate level phosphorylation? Yes, because I've got a phosphate transferred from a substrate to GDP to make GTP. I have a substrate level phosphorylation here. Please keep this in mind. It's the only reaction in the TCA where I have a substrate level phosphorylation. So it's the succinate thiokinase or succinyl-CoA synthetase reaction. That's a very important point. If you look at the enzyme, here's a picture of it. It has a Rossmann fold. There's your Rossmann fold right here. And what is the Rossmann fold involved in? If you remember, we learned this in chapter 6. The Rossmann fold is also called a dinucleotide binding fold. It's involved in binding nucleotides. And actually, the Rossmann fold in succinyl-CoA synthetase is involved in binding the ADP component of coenzyme A. Okay, that's just a little side note for you to know. Now let's look at the next reaction of the TCA. It's catalyzed by an enzyme called succinate dehydrogenase. Okay, what does a dehydrogenase mean? It's a redox enzyme. There's hydride transfer. Look for redox cofactors. But in this case, the redox cofactor is not an NAD plus or NADH, but it's really an FAD, which gets reduced to an FADH2. Okay, so, and in the process, succinate loses two H's and gets oxidized to fumarate, which has this trans double bond. Please make sure when you're drawing this that you draw the trans double bond shown. Don't draw the double bond as cis. So just one more thing to point out, here is FADH2, here is its FAD form. And what is it? It's a redox cofactor. We've actually seen this before when we were looking at cofactors in chapter 11. FAD can be reduced, an H minus can add here, an H can add here, and it can be reduced to FADH2. Just a little side note as well. Have you guys heard of vitamin B2 or riboflavin? What is it? It's this component, the blue and red component of FAD or FADH2. So that's if you take your riboflavin, it's good for you, okay, and it's good for healthy skin, healthy red blood cell production, etc. Just to point out that FAD requires riboflavin as a vitamin to make it up. Okay, let's look at the succinate dehydrogenase reaction. It's an oxidation involving FAD as your redox cofactor. Here's succinate. An H minus gets removed and transferred to FAD. An H plus gets transferred as well. I end up with FADH2 and fumarate. Please don't forget. So that's your hydride removal by FAD and a deprotonation. H plus and H minus both get added to the FAD right there. Also, don't forget that the fumarate formed has a trans, not cis double bond. Okay, so its double bond orientation is trans with respect to the groups attached to the double bond. Finally, please keep in mind, or here's something important, the enzyme is actually covalently bound to the FAD or FADH2 cofactors. There's actually a covalent bond between the enzyme via a histidine to the flavin group of this coenzyme, the FAD coenzyme. And so when it's in the FAD oxidized form or in the reduced form, both forms are covalently bound to the enzyme. One more thing to keep in mind. Succinate, here's succinate right here. It's a dicarboxylic acid that has 2 more carbons as well. So it's a 4-carbon carboxylic acid, dicarboxylic acid, excuse me. Here's malonate, a 3-carbon dicarboxylic acid. Can you oxidize malonate like you can oxidize succinate here? No, right? Because malonate has only 1 CH2. You can't make a double bond here. So malonate is a poison of succinate dehydrogenase. It's inhibited by malonate. Malonate is a reversible competitive inhibitor of succinate dehydrogenase because it resembles the substrate, it binds the active site, but it can't be metabolized in this manner by succinate dehydrogenase. There's one more thing to keep in mind. Succinate dehydrogenase is also really an enzyme of the electron transport chain. Okay? What happens is once electrons are transferred from succinate, from an H minus and an H plus to the FAD group of succinate dehydrogenase, and the enzyme gets reduced with its FAD cofactor, and it gets reduced to FADH2, it has now high energy electrons. These high energy electrons are transferred in the electron transport pathway to a ubiquinone, which is a molecule we'll look at and we'll see when we get to chapter 18 on the ETC. And the ubiquinone is reduced to ubiquinone H2, or its reduced form right here. And succinate dehydrogenase is actually a complex of the electron transfer chain or electron transport chain. It's actually complex 2 of the electron transport chain, or ETC for short. So please, very, very important, succinate dehydrogenase is really complex 2 of the ETC. Also very important, succinate dehydrogenase, because it's a part of the ETC, the enzymes of the ETC are found in the inner mitochondrial membranes. They're actually bound to the inner mitochondrial membrane. The complexes of the ETC are bound to the inner mitochondrial membrane. So succinate dehydrogenase, because it is complex 2 of the ETC, is also bound to the inner mitochondrial membrane. So please know, very, very important, succinate dehydrogenase, because it's a part of the ETC and the TCA as well, it's the only membrane-bound enzyme in the TCA. It's bound to the inner membrane of the TCA, of the inner mitochondrial membrane, whereas the rest of the TCA enzymes are in the mitochondrial matrix, as we've talked about before at the start of the TCA lecture notes. What happens in the ETC? Electrons are transferred from succinate via the FADH2 group of succinate dehydrogenase. Like we see, they go via this FADH2 group and they go through an iron sulfur cluster and they convert coenzyme Q or ubiquinone and they reduce it to UQH2. We'll look at that later when we get to chapter 18 in the ETC, but please note two electrons are transferred from succinate via the FADH2 group of succinate dehydrogenase to to ubiquinone, and eventually ubiquinone, and ultimately ubiquinone gets reduced to UQH2 from its UQ form. Oftentimes you can write these as just Q or QH2 instead of UQ and UQH2. The next reaction we're going to look at in the TCA, the second to last reaction, is catalyzed by an enzyme called fumarase, where fumarate is converted to L-malate. Okay, here's the reaction. It's actually hydration across a double bond. There's fumarate. You add OH to it, really, and you get this carbanion form. This is a reversible step, and then you add an H+, and you end up with malate. Okay, there's transitions of the elements of water and OH- and H+ across a double bond. So what kind of a thing is that? If you add, remember lyases? Yes, this is a lyase because lyases add water, ammonia, or carbon dioxide across double bonds or remove these elements to produce double bonds. Here, if you go in this direction, you're adding the elements of water across a double bond. So this is a lyase reaction. It's actually a reversible reaction, so you can actually remove the elements of water to make a double bond, and that also fits the definition of a lyase. And of course, this reaction is bidirectional. It can go in either direction because it's a lyase reaction, and lyase reactions are in general bidirectional. Okay, and the last reaction of the TCA is catalyzed by an enzyme called malate dehydrogenase. And this is the reason why you converted fumarate to L-malate, because L-malate has a secondary hydroxyl group. And what can you do to secondary hydroxyl groups? You can oxidize them. So there's still more energy to be found. I can still oxidize carbon, and this carbon, the secondary carbon with the secondary hydroxyl group, is oxidized to a carbonyl. And what can you do when you oxidize carbon? That's energy released, and you can harness that energy. You can harness that energy released in the oxidation of carbon to make a high energy compound like NADH. And this is exactly what happens. It's a dehydrogenase. It's malate dehydrogenase. What does a dehydrogenase mean? Redox reaction, look for redox cofactors, and look for hydride transfer. And that's exactly what happens. An H- here is transferred to NAD+ to give NADH, and that H comes off as H+, so I get L-malate oxidized to oxaloacetate, and I get NAD+ converted to NADH plus H+, and at the end of this reaction, step 8 in the TCA, I end up with oxaloacetate regenerated and ready for one more round of the citric acid cycle. So here's the malate dehydrogenase reaction, the last reaction of the TCA, reaction 8 of the TCA, in a little more detail. There's L-malate. What happens is you oxidize the secondary hydroxyl of L-malate. It's hydride transfer. The energy of the oxidation is captured by transferring that H- to NAD+ to give NADH, which has high energy electrons, which can be used to make ATP in oxidative phosphorylation, or the ETC and oxidative phosphorylation, which we'll get to in chapter 18. And you're oxidizing carbon, and that carbon gets oxidized to a carbonyl. That H goes as H- to NAD+ and gives you NADH, and that H comes off as H+, and you end up with oxaloacetate, and you can restart the TCA again. So let's look at a summary of the Krebs cycle, or TCA. One acetyl group through the cycle produces 2 CO2s. This is not really the acetyl group itself, but by stoichiometry you could say the 2 carbons of an acetyl group produce 2 CO2s, or are converted to 2 CO2s, though it's not the exact carbons that came from the acetyl group, but by stoichiometry you can consider them to have been converted to 2 CO2s. You get 2 CO2s, 1 GTP if you're a eukaryote, or ATP if you're a prokaryote, but GTP is interconvertible with ATP, and you get 4 reduced coenzymes. And which of these, or what do you get? You actually get 3 NADHs and 1 FADH2. So the acetyl group, you've got CH3 carbonyl connected to coenzyme A. It, in essence, you can say it's by stoichiometry is oxidized to 2 CO2s. But again, these carbons of the CO2s don't directly come from the acetyl group of acetyl-CoA. You get 3 NAD pluses being reduced to 3 NADHs, which have high energy electrons, plus 3 H pluses, and FAD getting reduced to FADH2, which also has high energy electrons. A GDP plus phosphate in a eukaryote getting converted to GTP, and this occurs by substrate level phosphorylation, and 2 waters as well are involved, and after the reaction that coenzyme A comes off, and you end up with free coenzyme A as well. So this is an overall summary of the reactions involved in the Krebs cycle, or TCA. One more thing to keep in mind about the Krebs cycle or TCA. It is amphibolic. What does that mean? It serves both in catabolic and anabolic pathways. And what are the anabolic pathways it's a part of? The citric acid cycle or Krebs cycle provides several intermediates for biosynthesis. For example, alpha-ketoglutarate can be used to make amino acids. Another example is succinyl-CoA can be used to make porphyrins. Another example is that fumarate and oxaloacetate, they can also be used to make amino acids, and amino acids, as you can see, can also be broken down into different intermediates in the TCA. For example, amino acids can be broken down into alpha-ketoglutarate. Citrate, you can see here, can be used to make cholesterol and fatty acids as well. Malate can be used to make gluten. And don't worry about this last statement about the citric acid cycle. The big thing to know or keep in mind is that it is amphibolic, and many biochemicals can be broken down into it. That's why it's called the hub of metabolism, so to speak, or it's considered the hub of metabolism because many biochemicals can be broken down into it, and the metabolites of the TCA serve as intermediates for the synthesis of many biochemical compounds as well.